going to pick up part 2 with objective 6, endo and exocytosis. These are both active transport, so requiring energy. The difference here is not just active transport like we saw before, these small particles. Endo and exocytosis involve transport of large particles, things that would not fit through the membrane no matter how big the protein. So endocytosis, really simple if you just remember endo into, exocytosis, just think of stuff exiting, going out of the cell. And we're going to watch an animation that I'm going to show you how to get there first. Instead of typing this all in, you might want to just click by going to my website, resource links page, and what you'll see when you click on, oops, let me show you where you're going, down here, I'll make these bold, endo and exo. This is the endo and exocytosis animation you want to watch for this. I'm just going to give you the abridged version. Some large particle that the cell needs to take into itself. So the membrane, you can see, just surrounds this particle and pinches off, now creating a vesicle or a vacuole, depending on what it contains. Alright, so the difference between solids and liquids we're not going to worry about, but there's a vesicle. Okay, so some vesicle made by taking in particles, and then when those particles are either broken down or maybe those particles are waste, need to get rid of them, same thing happens, only it's the opposite. That vesicle is going to fuse with the membrane and spit them out. The thing to remember here is the vesicle is made of phospholipids, the membrane is made of phospholipids and some proteins, but mostly phospholipids, so it's kind of just like two bubbles coming together. A smaller bubble bumping into a bigger bubble and just becoming part of the bigger bubble. And the opposite is true for endocytosis. Just kind of pinching off a little bubble from a big bubble. Okay, it's all made of the same stuff. That's how that's possible for this to happen. So if you want to click on that animation instead of typing all this in, that's how you get there. Resource links page, endo and exo. Objective 7, and again this is one of those ones you're going to want to focus some time on. Hypo, hyper, and isotonic solutions. Here's definitions, and it's very simple. Hypotonic causes water to move into the cell because the solute concentration is lower outside. Hypo is low, hypo, low, something you probably want to write down in your notes. Hyper, the opposite, high solute. Let's say this is water. So hypotonic would be like distilled water, no solutes. And hypertonic would be like the ocean, very salty, has a lot of solute. So in this case, water moves out of the cell in this case. Okay, and we'll get to these examples later, but something you want to maybe highlight. Water into for hypotonic conditions, water out of the cell in hypertonic conditions. And then isotonic, there is an equal movement in and out. There is basically no net movement is what we say. So equal concentration of solute inside and outside the cell. When you see some pictures, it'll hopefully make a lot more sense. So here's just a U-shaped tube. This is an old-fashioned U-tube. Hypotonic solution, you can see low solute or less solute than the other side. In between these two solutions is a semi-permeable membrane, something that allows water to move, but not the solute. Very important for this demonstration. Only the solutes can fit through these teeny tiny holes in the membrane. Okay, this is similar for the cell membrane, why I'm showing you this demonstration or this picture. So, as you can see, the solution levels, volumes, are equal. So if there's a lower concentration of solute here, there is a higher concentration of water on the left side. On the right side, there is more solute, a higher concentration of solute, so technically there is less water. This would be the example of like the ocean water compared to the water in your body. Alright, and then after time, what's going to happen is this, you can see here, the water is going to move from higher concentration to lower concentration. This is osmosis. movement of water across a membrane from high concentration to low concentration. So what happens? The solute particles don't fit through the membrane. They have to stay put, but the water moves. So the solution's volumes are going to change, but now the concentrations are equal. So this is isotonic, equal concentrations. If we were to zoom in to one teeny tiny spot here, you would see water moving in and out equally. Because on a microscopic, super microscopic scale, these concentrations along this membrane aren't going to be perfectly equal. So some might be a little higher on one side, a little lower on the other side. So the water is basically moving in and out equally, or back and forth equally. And like I said, this is a good example of, you're stranded out at sea. Why should you not drink the water? Well, here's the ocean water on the right. Here's your body on the left. If you start drinking ocean water, the water is going to leave your cells. You're going to become dehydrated, and eventually you're going to die. Objective 8 is about predicting the effects of hypertonic and hypotonic solutions. So let's say this beaker here is sitting in salt water, or is salt water. The little particles here are salt particles. On the inside we have a cell. That is a semi-permeable membrane. Again, the definition of semi-permeable membrane, simply it allows some things in and not others. In this case, we're going to say the salt is too big to fit through the tiny holes in this membrane. So, there's a higher concentration of water outside. When you see low concentration of solute, that means higher concentration of water. On the inside, a high concentration of solute, or also low concentration of water. So again, this is just osmosis. Water outside is going to move in because there's a higher concentration of water outside. This is the opposite to the example I just showed you. So in this case, water is going to go into the cell. Okay, so water is going from the beaker into the cell because there is more solute, or more importantly, less water, less concentration of water. The opposite is what happens to plants on the side of the road in the winter. When they're salting the roads because of ice and snow, a lot of plants on the side of the road are dying or withering up, and that is because of this term right here, plasmolysis, that I will get to on the next slide. So here's a picture of something that you should be able to see when we do this in class, but pretty simple. Here is a picture, two pictures, same picture but after time. Here's a picture of plant cells in a hypotonic solution or isotonic. So here you could put hypo or isotonic, but the clue here, the big deal here, is if you place those plant cells in hypertonic, say salt water. Salt water, there's going to be less water outside, more water inside. So once you place those in hypertonic condition, the water is going to leave the cell because there's less concentration of water outside. Again, always trying to find that balance, isotonic conditions. So in this case, you can see the cytoplasm shrinks up. Cytoplasm is mostly water. When the water starts going out, you're going to see it's going to get a darker color. It's like a higher concentration of solute in Kool-Aid. If you take the water out of Kool-Aid, it's going to get darker. Same thing's happening here. But technically what's happening is the cell membrane is literally detaching from the cell wall and kind of caving in on itself, trying to maintain the conditions that were over here. Alright, so this is called plasmolysis. Placing plant cells in hypertonic conditions. It's going to cause the water to leave. So you might want to put an arrow on your notes right here of water leaving, water leaving these cells. Okay, H2O going out. Now the rest of these notes are going to be blank. You're going to be doing some labs. So I'm going to show you some classroom demonstrations. You should have a good enough space, if not maybe use the back of some of these pages to draw anything that you need to draw, write anything you need to write from those labs and demos, because you will absolutely positively see those on the test. One more thing I wanted to show you. Again, just to kind of reiterate properties of this membrane. I don't know if you just heard that, but that guy was talking and he's annoying. So anyways, guys, notice the phospholipids. Here's the membrane, cell membrane. Outside the cell is what you're looking at. Inside the cell is below these guys. But notice how this plasma membrane, cell membrane, is very fluid. Okay, notice how those phospholipids are bumping into one another. Okay, there's space for tiny molecules to pass through here. But other molecules need to pass through using proteins that are spanning the membrane that are just kind of all over the place. So again, you can see these things just moving around. This cell membrane is very fluid. It's always moving. You can see the phospholipids are moving. There's lots of proteins here that are moving. Lipids, cholesterols, lots of stuff, but they're all moving around. And that's hopefully something to remember that will help you kind of get an idea, a really good idea of what this cell membrane is really like. So again, here's a good example of proteins that are attached to the membrane. Let me go back to this guy for a second. Proteins that are attached to the membrane or spanning the membrane, again, like this guy, is going to be allowing molecules, I paused it, that's why it's not moving, allowing molecules to pass into and out of the cell. So that pretty much wraps it up. Make sure that you have everything labeled that needs to be labeled, and pictures, definitions, highlight the vocabs, and don't give away your knowledge and highlighting and fill in the blanks to the slackers.